Let's see if I can do this. Just one moment, please. How's that?
Looks great.
Great. Well, thanks for that introduction, and thanks everybody for coming tonight, taking time out of your evening. So as Megan mentioned, before I dive into information about my research, I'm going to tell you a little bit more about PNNL. So PNNL is one of 17 US Department of Energy laboratories. Our main campus is located in the eastern part of Washington State in the Tri-Cities area. We also have campuses in Seattle and Portland, but one of the campuses I wanted to share with you tonight is the campus we have on the Olympic Peninsula. So PNNL actually has one of Department of Energy's only marine science laboratories, and that's located here in Sequim, Washington. This is where I work, located here on Sequim Bay. PNNL advances critical missions for the Department of Energy. Examples of this include promoting scientific discovery by operating national user facilities. These are facilities that are open to researchers from across the country. We are a national leader in battery development. We apply science and technology to develop solutions for national security issues. And we also develop novel solutions for environmental management, such as vitrification, which is a method for safely storing hazardous waste. PNNL has over 4,000 employees with a total annual spending of over $1 billion. Our intellectual assets include a number of inventions and patents and licenses. These data on the slide are specific to fiscal year 2019, but cumulatively each year these things really add up. In terms of our economic output for Washington State, we have a total economic output of over $1 billion. So in addition to our economic contributions to the state, Battelle and its employees contribute to the communities in which they live. So investments within the community during FY19 totaled over $500,000. A majority of those investments are STEM focused within our communities. Our employees volunteer over 30,000 hours within our local communities at 56 different organizations. So if you have some questions about these slides or the content of them, please let's maybe address these at the end. And with that, I will maybe dive into some of the research that we were going to talk about tonight. So I'm going to talk about habitat restoration in the Columbia River estuary. So I study biological communities in aquatic environments, and really the research that I'm going to share with you tonight is was focused on trying to understand how habitat restoration benefits juvenile salmon. So this map here on the left shows the extent of the Columbia River Basin, and the focal species that we were interested in was salmon. Salmon originating from watersheds across this basin have to transit through the estuary. So if you're not familiar, an estuary is really defined as the place where the river meets the sea. And so kind of an iconic photo of that is shown here on the upper right of the slide. Estuaries are incredibly important to juvenile salmon. So they don't just provide a habitat for fish that are originating nearby, they can essentially provide habitat for fish that are coming from long distances away. They provide places of refuge. They're incredibly productive habitats. So they provide a great food source for fish before they migrate out to the ocean. We were interested in understanding if restoration activities, so you see these excavators down here, so these would be activities that are intended to essentially restore habitats that might have been lost or disconnected. So interested in understanding if fish can actually benefit from them. So we'll get into that in just a minute here. So The Columbia River estuary provides a great place for salmon to be, but it turns out that humans have also come to rely on the areas within the Columbia and other estuaries, frankly, for that matter. In the Columbia, examples include things like Bonneville Dam. Settlement has typically occurred within these habitats. So here's a picture of Portland down here on the lower left. Shipping is a big industry within the Columbia. So the Corps of Engineers maintains a navigation channel so ships can get into ports throughout the river. Farming has historically and still continues to be prevalent in some places. This map right here shows the extent of the historic floodplain, and it also shows the areas that have been lost over time. So these shaded areas or the polygons within the map show all the areas that are essentially disconnected or no longer accessible to fish and other organisms. To address part of this, the Columbia Estuary Ecosystem Restoration Program, or SERP, was created by the Corps of Engineers and Bonneville Power Administration. The program began in the year 2000, and this is a timeline of different events relevant to that program. The thing that I really wanted to highlight here, though, is the adaptive management piece of this program. So adaptive management in its most simplest form is, I guess we could call it learning by doing. And so it's represented by these inner circles that you see here, these circles and arrows, I guess. So the idea here is that you would perform a restoration action, you would monitor or research that restoration action, you would learn, and then use that learning to inform additional restoration. In 2012, this is on this timeline here now, we had the SERP synthesis memo. And what that did is it actually sort of paused and gave a critical look at the program. So we evaluated whether or not the program was being effective. We decided to look at, what was going good, what was going bad, where were some of maybe the research gaps that still remained. And what was found with that effort is that there were a lot of really great things associated with the program. And there were also some major gaps still. And one of those gaps was the lack of understanding of how fish were actually deriving benefit from these habitats. In 2015, some researchers at PNNL got together with researchers at NOAA Fisheries, and we developed a conceptual model to formulate research questions and hypotheses. And this illustration is the output of that conceptual model. And really what it says here is that tidal wetlands potentially can provide both direct and indirect benefits to fish. So a direct benefit would be if a fish accesses the habitat, it's getting refuge, it's eating food that are being produced at the site. But that we also think that fish can have indirect benefits. So a fish doesn't necessarily need to enter into a wetland to receive those benefits. We think of food web enrichment as an indirect benefit. And the third piece to that was really trying to understand what the prey transport actually looked like from these systems. So we kind of divvied up what these tasks were, but the overall goal of this research really was to evaluate the ecological benefits of restoration action for juvenile salmon. Tonight, I'm just going to focus on the direct benefits aspect, or at least some of those components. Here's a map of our study area. So when I talk about the lower Columbia River and estuary, the extent of that, I think maybe you can see my pointer here, starts at the mouth of the river near Astoria, Oregon, and extends all the way to Bonneville Dam. So the tide actually can be detected this far away. It's about 234 river kilometers, I believe. So this is the extent of the lower river and estuary. We sampled within two different hydrological zones. So we had the upper estuary zone and the lower tidal river zone. And those zones are distinguished by differences in hydrology, as well as some other factors such as wetland condition and components. Our study included 8 tidal wetland sites. So we had 4 sites that were newly restored when we started our study. And so newly restored is really they were hydraulically reconnected to the floodplain. In one case, we had a habitat that was newly created habitat. And the design relied on a paired study design. And so what that means is that for every restoration site, we matched it up with a paired reference site. And that ended up being really important to our study because it really helped us benchmark some of the patterns and trends that we were seeing at these restoration locations. So what do these locations look like? This is one of our restoration sites called Carlson Island. Carlson Island has an interesting story. Historically, it was developed by a family, the Carlson family, and used for farming. So they, this family built a house on the island. They built this lovely network or perimeter of dikes. So you can see these lines here in the photo along the outer edge of the island, and then you can kind of see this this nice little square located in here. So this is surrounded by dikes. There's also ditches that were dug for drainage. The restoration effort, so eventually what happened is the conditions were too harsh, and the family decided to abandon this island, and the land was later transferred over into a refuge. And when restoration occurred, the big focus of restoration was to remove these tidal dikes and encourage reconnection of habitat. So you might be able to see here on the photo these breaks in what appears the dike. So that would have been where the excavator came in and removed fill. Another one of our restoration sites occurred at Steamboat. So Steamboat is part of the Julia Butler Hansen Wildlife Refuge, where they provide habitat for Columbia white-tailed deer. So this refuge, there's a road that went across the river. This is the main channel of the Columbia River. So it's a really popular place to go fishing. It still is to this day. But what ended up happening is the road started to fail. So all the fill material that was protecting this was beginning to erode away because of the Columbia, and it became a little problematic. No one quite knew who was responsible for fixing the road. And certainly this dike was really important because it prevented flooding from happening for the deer. So the Corps of Engineers saw an opportunity, and what they did is they came in and they installed a setback levee beyond the main channel, and then between the levee and the main channel, they actually dug new channels. So this is a habitat creation project. This is an aerial image of the site in 2012. This is the outcome of their judging and excavation effort. So these are tidal channels. This main channel right here maintains water during most of the tides, but some of these distributary channels will fill and drain with each tidal cycle. This is an aerial image the following year. This is in 2017. So our job really was to understand how these newly restored habitats were actually functioning. So how fish were using these systems. We were interested in understanding, what fish looked like in these areas. What was their condition? And I'll get into how we measure that. So I mentioned the focus of our study was fish. So those are biological communities. But if you're wondering what a day in the life of a fish biologist looks like, here it is. So we're doing a lot more than just measuring fish and biological communities. We look at how the wetland channels are shaped over time. We collect channel cross sections. We measure sedimentation on top of the marsh surface. And all this information provides a better picture for the overall function of these systems. In wetland channels, our kind of go-to method for capturing fish is what's called a fyke net or a channel trap net. And the way that this system works, it's called a passive gear. So you would enter into a wetland channel at a high tide and set your net across the entire channel right here. And then you can let the tide essentially do the fishing for you. So as the tide is receding, the water level is decreasing, it's going out of the channel, and the fish are therefore also going out. There's a live box in the net, so we can sit at the live box and sample fish as they enter in. And then we work up those fish and collect information and data in a number of ways. In this channel, you can see we actually have another net over to the right, and so that was just really a block net. We didn't want any fish escaping out the back door, so to speak. One of the downsides to this gear is that at the end of the day, you can have a pretty long and muddy walk in order to get home. And you hope that by the time you get to the end of that channel, that the boat is where you left it. We haven't been abandoned by a boat, but sometimes if you get to the end of the channel, you can also still have a low tide and you need to wait a little bit longer for the water to come back in. In addition to these fyke nets or trap nets, we also use a more active gear type. These are called beach seines. So some of the channels are pretty small, so we have to, basically size our net to the size of the channel. So some nets can just be pulled with 2 people. Some nets are so big that we need boats to deploy them, and then a lot of strength to haul them in. The one thing that we use beach seines for, in addition to fyke net, is that it actually allowed us to capture fish. So we would set up a fyke net, and then we could go capture fish with a beach seine and mark those fish and release them above the net. And then it helps us actually estimate the efficiency of our gear. So we have a better idea when we do things like extrapolate density estimates for salmon, for example, in these tidal systems. I mentioned working up of the fish. So for the most part, we capture and release fish. We do take some fish for additional analyses. But what we're doing typically is we're identifying fish to species level. We collect information on length and weight of fish. We sometimes will take a tissue sample for a genetic stock analysis. And the other thing that we really rely on is gut content analysis for a lot of our data sets. And so this photo here is a picture of a fish undergoing what's called gastric lavage. So we use a syringe to insert some water and essentially flush its gut contents into this funnel. We then take that, it's called sometimes a bag of science, as one colleague puts it. So we take that data or information back to the laboratory and we analyze it. And so these are photos here of things that we might see in the gut contents of fish. Okay, I'm going to now go through some slides of data that we have collected. But before I talked about sort of the biological data, I did want to talk about river discharge and temperature. So we have 2 panels here. The one on the left is discharge. The one on the right is temperature through time. The gray shaded areas in this plot are the 10-year running average. So in 2016, when we conducted our study, there were a couple of points that were maybe a little above the 10-year average. But in 2017, it was a completely different story, as you can see here. So we had these really high peaks, high discharge in 2017 compared to 2016, but also much higher than the 10-year average. So this is not a desirable condition when, from a study design perspective, and that's especially true when you only have a 2-year study to conduct. So we made the best of it. We had to, kind of do some creative modifications along the fly. But you'll see this come up in some of the data that I talk about later. The temperature, the point here with the temperature, so we saw a lot more water in 2017 than we did in 2016. And we also saw slightly lower temperatures as well too. Okay, so these data slides that I'm going to kind of cruise through here. I just wanted to note that we use a number of different modeling techniques to evaluate our data. So I'm going to gloss over, I think, the details of those, but if you have any burning questions about the analysis, we can maybe discuss that at the end here. So because of the different conditions in water years, we were really only allowed to derive density estimates for Chinook salmon in 2016. And the reason for that is that the fyke nets for some locations and some time were just ineffective. So I mentioned, we really rely on the water funneling the fish into the channel, but what happened in 2017 is that most of these channels under normal conditions drain almost completely. You saw a photo of that, right, of somebody walking out of the channel at low tide. In 2017, that was not the case. There was a tremendous amount of water still left in the system, and so it just meant that our fyke nets were we were not getting the estimates that we thought were reliable, and therefore we couldn't calculate density. So here we have it, 2016 density estimates. So we use a linear model to fit density, and we use a number of explanatory variables to help us understand which of those variables most explains density. When we asked the question about, whether or not density of fish was greater in reference sites versus restoration sites, we do have an indication that there is a slightly higher density at reference locations. But what we found in our analysis is it's not necessarily true across the board. And so it's really important that we carefully kind of consider each one of these sites on an individual basis, and maybe not make such broad sweeping statements about overall condition of restoration sites. The exception to that was, I'll give you the example here. So Steamboat, that site that I showed you, was a creation site right next to the main channel of the river. So this is the line, the density line for Steamboat. So this is the density. Its corresponding reference location is down here. So this is this Welch Island is its pair. So Steamboat had more fish, even though it was a restoration site. The other restoration sites did not follow that trend. So genetic stocks are an important factor from a fisheries management perspective. So not all stocks are threatened and endangered, and fisheries managers tend to be particularly interested in certain stock groups. So that's why we included this data in our study. I guess the things to take home here are, so we have a couple of columns here. So on the left, these are all the reference sites. On the right are the restoration sites. So these are the 5 different genetic stock groups of Chinook salmon that we encounter at our site. So you can see that this West Cascade Fall, so these are the green bars, these are the most predominant stock group that we tend to encounter, with a little bit of variation from other stock groups as well. We use a multivariate analysis to ask questions about whether or not we see differences in genetic stocks by location or type or month or year. And really what we found with this analysis is that most of the variation in the genetic stock composition actually comes from season and interannual variation. We found that space was not a very good explainer of genetic stock diversity within these sites. So we didn't see any differences between the hydrologic zones, and we also didn't see any differences between whether or not a site was restored or not. Next, we considered fish condition. So, really understanding the health and condition of these fish is a pretty important thing. We used a relationship between weight and length of a fish. So what we do is we do a linear regression, basically. So we have length along the x-axis here, and along the y-axis we have weight. And what we saw was a little surprising. We saw a remarkable similar pattern across all of these sites and across time. So there's a really tight fit between length and weight. But I think what was more surprising is that we saw such consistency in this relationship across all these locations. Diet biomass. Okay, so I showed you guys the slide of gastric lavage. So we do a couple of things with diet data. One is we can look at the overall biomass. And so all that really means is how much do the gut contents weigh? And the weight of that is an indication of, essentially how much food fish are getting. The other thing that we look at is the community composition, or composition rather, of the gut content items. And I'll show you some slides on that in a minute here. But the biomass here, what we have along the y-axis, so this is the this vertical axis up and down, we have the biomass, and along the x-axis we have the Julian date. So each one of these squares here compares a reference and restoration site pair. So at this upper left panel here, we have the Battwater Crims pair. So Battwater, in this case, is the restoration site. And we see that, so this is what this is, this light gray circle here. So the biomass at Battwater was quite a bit lower than the biomass at Crims. This is also on the log scale. So this is a pretty substantial difference. We saw the same was true for those two site pairs in 2017 as well. You know, when we look at the Dibley-Fisher pair, we see that maybe there's some more similarities, but in 2017, we do begin to see, at least for certain times of the year, that there were some major differences in the total biomass. The pattern maybe becomes a little bit less distinct as we look at some of the other restoration reference site pairs. But on a whole, when we analyze these data, we do come away with a message that there are some differences in biomass, that the gut contents of fish tend to be fuller when sampled from reference sites as opposed to those newly restoring wetlands. Okay, Chinook salmon diets. I'm gonna orient you on what the slide is and how to interpret it, because you're gonna see it again in a few more slides. So we use what's called a joint species distribution model to look at variation in the diets of Chinook salmon. And so this list of organisms that you see here includes individual organisms found in the gut contents of fish. So I believe there are 19 of them. This is not the full list. So for the analysis, we choose the lists that occur essentially in 25% of the samples or more. So one of the things that we can know from this is that fish are eating a diversity of prey organisms. These little letters here after the name indicate different life stages. So an A means adult. And so if you look at all the A's, those correspond to insects. So insects are a favorite food of juvenile salmon. Some of you might know that. P stands for pupa. And N is not applicable. So an Ecstuary would be something like a mite. So, okay, this is the list. So these are the organisms, and then we have these little lines right here. And essentially what these are, we think of these as effect sizes. So we look at this line right here. The dark bars are the 50% credible interval, and the light bars are called the 95% credible interval. And so what this really does is just helps you understand the spread of the data and the effect size. This vertical line right here is the 0 line, and this helps us orient on the graph. And so what we can say here is that if a value falls to the right of this 0 line, there are more of these organisms in the restoration site. So if one of these values falls to the left of the 0, there tend to be more of these in the diets of the fish at the reference site. Okay. So now that we're oriented, what do we say with this graph? So if we're asking a question of whether or not there's a difference in diets of fish between reference and restoration sites, we say that yes, there are a difference in diets. So we see more copepods and more cladocerans, more columbullans in the diets of fish found in the newly restored site. We see more chironomids, both adults and pupa, in the reference site. So chironomids are a special favorite food of juvenile salmon. Chironomids are a relative of mosquitoes, but they are the non-biting form of mosquitoes. So they're midges is what they are, if you're familiar with those. So yes, there are differences for some of these taxa, but look at this. There are a whole lot of similarities too. So anytime we see these bars falling on the line, that means that there's overlap. So essentially there's no difference, that these things occurred in the diets of fish more or less equally between both restoration and reference sites. Okay. So we are really focused on understanding restoration versus reference, but I wanted to share this with you to provide a little bit more of a fuller picture. So this is not there's more to the story than just whether or not a site is restored or not. So a couple of things that we found in our research. One is we see differences across the year in the diets of fish. So interestingly enough, in 2017, we found that juvenile salmon were eating many more amphipods. So you see that right in this panel that's located on the right. And we saw more amphipods in 2017. We also saw that more of them in the estuary zone as opposed to the tidal river zone. Amphipods are important note because amphipods can be kind of large. They can be juicy and nutritious. And the fact that we saw more of them in 2017 and that they're more benthic orienting causes us to think that there could be some interaction there with flow and food quality. But the other thing that we see here in these figures is that location matters. So we did see a trend for differences in diets depending on where we were in the estuary. And maybe that's not particularly surprising. The Columbia River gradient is huge. We noted that it's over 200 river kilometers long. There's a lot of different environmental conditions across that gradient. But this is really important from a restoration planning perspective. So it provides some insights into landscape scale design and principles. Okay, this is the last time you have to look at this slide, I promise. So foraging opportunity, up to now I've been talking about fish. So we were really focusing all our efforts on Chinook salmon in particular. We were interested in knowing, what the fish are eating, but the other aspect of that is what's available to the fish. And so we wanted to understand foraging opportunity. And to do that, we looked at various prey communities that are being produced in these habitats. Salmon are really interesting. Salmon will feed on a variety of things in these habitats, as we saw from the gut content analysis, but they also feed from different, what we call prey pools. So salmon will feed from sources that are relevant to the marsh surface. So remember those adult insects, right? Winged organisms. So salmon are collecting those at the water surface, and those have more of an interaction with what's happening on top of the tidal marsh. Fish also feed in the water column, so they can collect organisms, invertebrates that are suspended within the water. And then the benthic environment. So the benthic environment is essentially the mud, if you will, within the tidal channel. And certainly as fish get larger, they can exploit those benthic prey resources more. But the benthic environment is an interesting environment in that it actually tends to be the sort of the original source or producer, if you will, of some of these insects. So as adults, insects are flying around, but as larvae, they can be more associated with the benthos. So each of those three prey pools were important for our consideration. And what was interesting in these findings, and you can already see this here, is there's a lot of variability. You see these bars that there's a lot of spread in the data associated with the marsh surface. We also see that there's a lot of spread in the data with the invertebrates collected within the water column. You'll know if you kind of sort of painstakingly try to compare the list too, you'll notice that the lists are not similar, so that you can get a different community composition of things that are in the benthic environment versus a different composition of things that maybe we captured near the marsh surface. So we don't see a lot of differences between reference and restoration sites when we only look at prey communities on the marsh surface or prey communities in the water column. We do see some segregation in the communities of invertebrates that exist when we do look at the benthic environment. And so what this tells us is that, especially for future studies, it's really important to sort of to be able to map out what your hypotheses are and really formulate those research questions. So for example, if we were interested in foraging opportunity and we know that fish eat adult insects, we might tend to want to evaluate insect communities at the marsh surface. If we did that, we might have walked away with some conclusions that led us to believe there really were no differences in restoration and reference sites, and they're not from these data. But the full picture, I think, comes into play when we consider these other communities as well. Okay, so a lot of that, the discussion about community composition. So we talked about, there's a number of organisms that salmon are feeding on. They're diverse. They vary in terms of their density and their abundance. But there is this element of the so what part, like what does that all mean? So there were, 10 more organisms at the site than the site. Like what do we do with that data? How do we use it to interpret the ecological significance of that? One of the things that we were interested in doing is really examining the quality of the prey that are available to fish and trying to frame that question around whether or not we see differences in quality between what would be newly restored sites or sites that are reference sites. So, reference sites would be these locations that are more mature and presumably are functioning differently ecologically. So the way that we do that is energy. We can look at energy content much in the way that you might look at energy content of the food that you eat, right? So maybe you count calories or look at carbohydrates. So we can do the same thing with invertebrates, believe it or not. And so for this, we actually went out to the literature, and so there's some existing information out there about these various organisms and how many calories they are, or kilojoules, if you will. And we used those data to model what we found in the gut contents relative to the weight of those items, and then began to ask some questions about whether or not we saw differences in space and time. And the results of that are displayed here, and you probably already see that there's a lot of overlap in these bars. So the y-axis here are essentially the energy density values of the combined prey organisms that we find in the gut contents of fish. And along the x-axis, we have these locations. Each one of these panels is a different month. And so we see a lot of overlap, but there are some places where we do see quite a bit of difference. So for example, in May of 2017, we see that the reference site had a much higher energy content, or at least the gut contents did, than the restoration site. And so that really allows us to dive into those data and understand what's going on. That's helpful from the perspective of really being able to inform maybe some of the mechanisms that are driving some of these processes. Okay, no more data slides. I'm going to show you the summary slide here. So what have we learned in all of this? We heard a lot. There was a lot of data collected, a lot of different information. What we found out is that juvenile salmon are, in fact, using these newly restored tidal wetland channels. We found that differences in genetic stock composition of Chinook salmon were not really explained by site type, but were more explained by month and year. We saw that fish condition was remarkably similar across sites and years. In terms of the gut content analysis, we found juvenile salmon are eating a diversity of prey items. They're doing that both in these newly restored sites as well as the reference sites. We did find some evidence of differences between those site types, right? So we saw higher biomass of gut contents from reference sites. We also saw in some cases, fish were eating if we want to call it their preferred prey organism, chironomids, so more chironomids and reference sites, but they were eating a lot of things in their restoration sites as well too. We found through the diet analysis that location really matters. So fish are experiencing different habitats depending on where they are along that gradient. And that's really important to provide information for restoration planning. Maybe really trying to hone in on locations where you can get the most bang for your buck in terms of providing good opportunities for foraging for fish along that gradient. In terms of those foraging opportunities, we found that not all prey pools will be good indicators of site scale patterns. So, if a research project is really interested in understanding site scalability, then, some really careful consideration needs to be applied to asking, which prey pools are important for that. And then finally, the energy density from these prey resources was really similar across the restoration and reference sites. We saw a few cases where there were some differences, and those provide some indication for us to, go back into the data and take a look and try to understand, were fish maybe consuming a lot of organisms that just are not particularly energy rich, if you will. And why is that? I am incredibly grateful to all of my colleagues who have contributed to the study. As you can see here from this huge list of people, it really does take a village. Our research for this project was funded by the Army Corps of Engineers, Portland District. And our team is, really comprised of just a diverse, incredibly dedicated set of people. So thank you to everyone who contributes. So that concludes my discussion for tonight. As Megan mentioned at the beginning, please fill the survey out. It helps to inform future presentations like this, and we really appreciate the feedback. So
thank you, Nikki.
Yeah, with that, thank you.
We appreciate you taking the time to discuss your research tonight, and I know the audience appreciates it as well. We had 37 people joining us tonight, and they do have some questions for you. So if it's all right, I'll go ahead and jump right in with the Q&A portion.
Sure.
So early on, we received a couple of questions. What factors are incorporated in fish density models?
Oh, yeah. So the fish density, well, I guess a couple of things. One is how we kind of estimate fish density with the field data. So the way that we do that is we look at the number of fish that we capture in our net. We also use, I mentioned efficiency estimates, right? So we can then, I guess, extrapolate based on those density estimates and the efficiency estimates combined, and we divide that over the total area that we sample. So we calculate the total channel area above the net. So that's kind of the basic form. The models themselves use things like month, they use sampling occasion, we use year. I think site pair is another factor. And the other thing that we use is it's called an interaction term. So all of those variables in the model can then be, you can combine variables together for an interaction term. And so we did that, and the interaction terms were really the kind of the indicator for us that all the site pairs aren't behaving similarly but through space and time.
Thank you. Why is it surprising that length and weight are strongly correlated?
That is a great question. I think it has to do with how fish are growing, right? I think the thing that so we do see this, right? We see anomalies, and you saw that with the data that maybe some fish are longer and don't weigh so much, or whereas some fish tend to be not quite as long and a little bit more round. But really, I think it just has to do with the growth trajectory of the fish. I think the surprising thing to us is to see that repetition through space and time. And that is something that's a little bit perplexing to me. We have another data set, a study from the Columbia River, where we looked at genetic stock composition through time. And again, we were surprised when we started amassing this years of data at the consistency in the appearance and time of the stock groups. So I think a lot of this also relates to sort of inherent genetic traits in these organisms too, and how these manifest then in terms of growth conditions.
Great. Do you anticipate that as restoration sites proceed through ecological succession, you'll begin to see fewer differences with reference sites?
We think that possibly, yes. Like the $1 million question is when? When does that happen? There's, in terms of restoration ecology, it could be all, it depends, right? So oftentimes restoration programs like SERP, when they implement adaptive management and monitoring, they will do things like monitor for the first 2 years after, and then go back at year 3 and go back at year 5 and then year 10. And then that gives a good indication of sort of the trajectory of time and how these systems basically equilibrate. So yes, that is the hope, that they would become more similar to reference location.
And would that be the same with the diet composition? Do you expect that those would become similar over time?
Diet, perhaps, yes. And I guess I would say that the differences that we saw in diets, I don't know that I would qualify them as substantive. So certainly there were differences, and certainly those differences were not too terribly surprising. But yes, I would suspect that those would kind of come to equilibrium much sooner. Things that we would look for changes over time in these systems would be the channels themselves and how they form and deposition, and maybe not so much the biological characteristics of these habitats, but the physical characteristics.
Okay. And does the difference in the diet give insight to the health of the fish? Are some more nutritious than others, or is it just an indication that the restoration has changed what types of foods the environment can provide?
Yeah. Yes. So I mentioned to you some of the sites in the lower tidal river had more zooplankton. So we mentioned copepods and cladocerans. So those things maybe would be less nutritious and meaty. So like if you could think of it like the human equivalent would be, would you get more nutritional value if you ate an entire bowl of popcorn, or would you get more nutritional value if you ate a steak? And so that's really kind of what we're talking about here. So some of these organisms, the invertebrates, are so incredibly abundant. So I think one colleague says, you know, salmon are literally swimming in copepods. So, you know, it would be hard not to eat them. And you know, while there are a lot of them in the system, and maybe they don't cost a lot of energy to capture, they're not necessarily nutritionally valuable, like say an amphipod for a big juicy amphipod.
Are the ages of the restoration sites similar, or were they created at different times? If the latter, did you see any convergence with older sites in the restoration areas?
That is a great question. And that's something that, so we just are closing out on this research. So we've just completed our final report and submitted that. And that was one of the questions that we also addressed during our review period. The answer to that is the restoration sites are different in age. So anywhere from 1 to 3 years, I believe, is how old the site was, the newly restored site, when we started the study. You know, on the one hand, you know, we had 8 sites and 4 restoration reference sites pairs, and that's a lot. But it's also not from kind of an analytical perspective, like that's pretty limited in scope. And so our ability to kind of tease out how the age of the restoration site impacted some of the patterns is, we'd be pretty marginalized, I think, if we tried to kind of tease some of those things out.
Okay. Do you have any suggestions for where and when to see salmon runs near Squim?
Oh my gosh. No, I don't. I think I would probably open it up to the audience and see if anybody, you know, so as a researcher, my focus has really been on juvenile salmon, but I cannot tell you how many times where, you know, being at a boat launch with all the gear, you know, people are curious and want to know what we're up to. But one of the very first questions I always get is, where's the best place to catch fish? And I have to say, I only know about juvenile salmon. I'm so sorry.
Let's see. With time, the restoration sites are likely to become more, oh, nope, we talked about that. More natural. Is the food content likely to become more similar? Will studies like this help inform management of dams as far as when water should be spilled for fish?
Oh. You know, perhaps. I think there's a whole lot of information that goes into fish spill and mitigation. certainly, so spill, for anybody who doesn't know, would be in reference to the dams, the hydropower dams that exist on the river. Spills, definitely a hot topic, and it certainly relates to overall availability of habitat. I would say that, just like the cautious response in that would be, certainly the study wasn't designed to look at that, but, there's a seasonality component to the research that I think that you could begin to kind of tease some of those things out. How spill might relate to things like temperature and inundation. You could try to take a look at some of those things.
Thank you. And how are other organizations using your findings to inform their work, like conservation or restoration organizations?
Yeah, there are, so I mentioned the SERP program. So certainly the main, what we call the action agencies are BPA, Bonneville Power Administration, and the U.S. Army Corps of Engineers. So they use this effectiveness research to make decisions about restoration and prioritization. The program, to my knowledge, is pretty focused on restoring and maybe not so focused on conservation efforts. So the difference between restoration and conservation would be, although maybe somebody might argue about this difference, at least in the Columbia River, restoration is, sort of acquiring lands and performing some sort of action to, make it hydrologically reconnected. There are some local groups in the estuary that use this data, or not necessarily just our data too, but all data to help things like prioritization, I think is probably the most appropriate way to describe that.
Okay. And we had a question about, is there published literature on this?
Yes, well, so this study actually, so I just mentioned that we finished our report. So we've submitted the report to our funder, the Army Corps of Engineers. And so the next step is to publish the results of the study. I also mentioned our collaborators at NOAA Fisheries. So the research that I shared tonight was sort of one component of two other components of the study. And so there will be a lot of information coming out about this probably in the next year or so.
In your acknowledgement slide, you included a category called isotopes. How are isotopes involved in your work?
I didn't talk about the isotopes tonight. There were a lot of things I didn't talk about that we did for this research. So we used isotopes a couple of ways. One, in the direct benefits aspect, we were interested in understanding if we saw a seasonal change. And so isotopes, so I should back up, we looked at carbon and nitrogen. We collected fin tissue, and then we performed isotope analysis on the fin tissue. And so the isotopes help to provide us a picture of previous experiences by the fish, so like up to 6 weeks. So fish are assimilating signals based on the food that they're eating, and then that helps provide us an indication of maybe where the fish has been and what it's been up to, so to speak. So with the direct benefits research, we were interested in looking at site scale variation as well as temporal effects. But then we also combined this isotope research with the research from our colleagues at NOAA Fisheries to understand how different fish are using different habitats. And in that case, the isotopes provided a really fascinating story about fish distribution across the estuary. That will be another talk for another day.
Perfect. So I think we have time for one last question, and we'll end it with this. Did you learn anything new or unexpected?
Oh, I think, yes, we are always learning things new or unexpected. I mentioned the 2016, 2017 water year. That was a big learning for us. I think probably the biggest thing that was interesting for me was really looking at the foraging opportunities of fish and understanding how it's really important to to carefully select what would be your monitored indicators. I think another thing that was related to that of interest is really being able to look at the patterns and trends that we see and take it a little bit one step further. So, you know, being interested in looking at prey quality as opposed to just the quantity and types of organisms that we encountered there. I think we started to kind of just scratch the surface on that, and we're looking forward to digging into that a little bit more in the future.
Great. And we did have one follow-up on the isotopes. It was the locations have different isotopic signatures. Is that correct?
Yes and no, and it depended on which part of the research objective we're talking about. So in the study that I didn't talk about, what we did basically is that we looked at fish that were migrating in the main stem, and then we looked at fish that were occupying these tidal channels. And so those were Chinook salmon. And what we found is that even though they were the same species, they had, and in some cases, even though they were the same genetic stock group, they had very different experiences as indicated from their stable isotopes. And so what we said from that is that fish in the main stem that were, you know, potentially deriving these indirect benefits were different or had a different feeding history than the fish that we captured in the wetland channels. Again, even though there were same stocks. And so that actually was really pretty surprising for all of us. Yeah, I mean, I could talk more about that, and maybe I can follow up too if somebody wants to have a specific question.
I'm going to say we're going to have to schedule you for another seminar. Well, thank you, Nikki. We appreciate you taking time out of your day to share your research with us in the community. And thank you to everyone for attending tonight's seminar. We really appreciate you joining us tonight. Just a reminder one last time to take our brief survey. We'll have the survey link in the chat window. There it is. You could also sign up for future seminars at the end of the survey. Again, thank you so much to everyone who joined us tonight. We hope to see you when we have more seminars scheduled later this summer. Everybody have a great evening.
Thank you.